Hello my dear students, welcome back. So in the last video we studied about the plastids. Okay. Now in this video we are going to start another cell organelle, that is ribosome. About ribosome I discussed in the prokaryotic cell, in mitochondria and in chloroplast also. Okay, so it is not something new cell organelle for you. About this some points already I discussed to you. Okay, so first thing you should know that the name of scientist who discovered, or we can say who observed this ribosomes. So the name of scientist is George Palade. So George Palade was the first scientist to observe the ribosomes. Second thing is that these ribosomes, as I told you earlier also in the case of prokaryotic cell, so it is a cell organelle which is not bound by any membrane. So it is a membraneless cell organelle, means it is not surrounded by any membrane. Clear? Then you may be asking how it is present if it is not surrounded by any membrane. Actually, it is present in the form of granules. It is present in the form of granules. Okay, so it is present in the form of granules inside the cell. And this is the cell organelle, which is also called as organelle within a cell organelle, because it is lie freely into the cytoplasm, and apart from the cytoplasm it is also present inside the three other cell organelles like mitochondria, chloroplast, and also on the endoplasmic reticulum. If you remember, in the endoplasmic reticulum I told you about the rough endoplasmic reticulum. So in the case of rough endoplasmic reticulum, they have the deposition of the ribosomes, means they have the ribosomes over the surface. That's why it gave the rough appearance, and hence the name was given rough endoplasmic reticulum. Means we can say this is a cell organelle which is not only present inside the cytoplasm, but it is also present in other cell organelle. That's why it is called as cell organelle within a cell organelle. Clear? Then about this structure I will discuss. So first you write down some points. So George Palade was the first scientist to observe ribosomes. So name of scientist here is very important. Many times it is asked in the entrance examination also. Clear? So ribosomes are membraneless cell organelle which are present in the form of dense particles. So they are present in the form of dense particles. Clear? One more thing you should know, that what actually they composed of, what is the chemical nature of the ribosomes. So these ribosomes, they are composed of RNA and protein. Very important. So the chemical nature of the ribosome is ribonucleic acid, means we can say RNA and protein. So ribosome are composed of RNA and protein. Clear? Then what is the function of ribosome? So the main function of the ribosome is the protein synthesis. So this is the reason that they composed of protein and RNA. How the protein synthesis take place inside the ribosomes, that I will discuss in the class 12th. Okay, so a topic is there, translation. Under that topic it is taught how the protein synthesis take place inside our cell. Clear? So the function of the ribosome is in the protein synthesis. So ribosomes are concerned with the protein synthesis. They are concerned with the protein synthesis. So they are concerned with the protein synthesis. Clear? Now we will see their structure. So even if you see the structure of ribosome, here a simple diagram I am making here. So usually if you observe the structure of the ribosome, you will find that ribosomes are composed of two subunits. Ribosomes are of two types that already discussed to you, 70S and 80S. So I told you that ribosomes are of two types, 70S and 80S. 70S is mainly the prokaryotic ribosome and the 80S is the eukaryotic ribosome. So whether it is 70S or 80S, all the ribosomes composed of two subunits. So this is the larger subunit, and the smaller subunit. So two subunits are there, okay, larger subunit and the smaller subunit. So I told you that whether it is prokaryotic ribosome or eukaryotic ribosome, they will have the two subunits. And what is the significance of these two subunits? That also will study in class 12th, because this type of structure is required for the protein synthesis. Okay, but here just you should know that the ribosomes are composed of two subunits, larger subunits and the smaller subunits. Clear? So the prokaryotic ribosome is 70S and eukaryotic ribosome is 80S. Now here I will tell you what is the meaning of S. Actually S stands for Swedberg unit, or we can say it is sedimentation coefficient. And this Swedberg unit or sedimentation coefficient, it is the measure of density and size. It is the measure of density and size. So in a note you write down here. In ribosome, S stands for Swedberg unit or sedimentation coefficient. Which is a measure of density and size. Very important. Which is a measure of density and size. Means if we conclude, the prokaryotic ribosome is smaller because it has the 70S, and while the eukaryotic ribosome is larger because means it will have a larger size as well as density because it is of 80S. Now just now I told you these ribosomes are composed of two subunits. So this prokaryotic 70S ribosome is composed of 50S and 30S. Means the larger subunit will be of 50S and the smaller subunit will be of 30S. Don't simply add them and say that 50 plus 30 is equal to 80. It is not mathematics, okay? So combinedly these two, they are composed of 70S, okay? But the larger subunit will have a density of 50S and means 50 Swedberg unit, and the smaller subunit will have a density of 30 subunits, 30S means 30 Swedberg units. Clear? And similarly in the case of eukaryotes also, so as they have the 80S ribosome, so the subunits are 60S, the largest subunit will be of 60S and the smaller subunit will be of 40S. So here also you can see if you are going to add them simply, then it will be 100S, but in reality it is 80S. So eukaryotic ribosome is composed of 60S 60s in bracket we made on larger subunit, means larger subunit will be of 60s, and 40s it will be smaller subunit. Okay, so this also you should know. Now, one more point about the ribosome we write down. I told you that ribosomes are the cell organelles which are not found freely only in the cytoplasm, but also they are found inside the other cell organelles. That's why these ribosomes are also called as organelle within a cell organelle, because apart from the cytoplasm they are present in other cell organelles also. So ribosomes are called organelle within a cell organelle, organelle within a cell organelle, because it is present in cytoplasm as well as in other cell organelle, and that example also I told you that what are the other cell organelle in which this ribosomes are present. That example like endoplasmic reticulum. That's why the endoplasmic reticulum which have this ribosome they are called as rough endoplasmic reticulum. Then mitochondria and chloroplast. In the last video you've seen this one, in the last couple of videos, that mitochondria and chloroplast also they have the ribosome, even though that ribosome is of prokaryotic type, means they have the 70 years. Okay, so this is all about the ribosome here. Now our next discussion is cilia and flagella. Cilia and flagella. Cilia and flagella. So both these two, means either it is cilia or flagella, they are the extensions of the cell membrane. Actually, this cilia and flagella they are studying about the eukaryotic cell. So in the case of eukaryotic cell, this cilia and flagella they are the extensions of the cell membrane. While in the case of prokaryotes, we have seen that this flagella was the extension of the cell wall. So this is a major difference. Okay, so in the case of prokaryotic cell, the flagella is the extension of the cell wall, while in the case of eukaryotic cell, the cilia and flagella both they are the extension, or you can say they are the outgrowth of the cell membrane, and their function is also same. Both of them are concerned with the mobility, or we can say they are concerned with the locomotion. Okay, means we can say movement. Different words are used in the different words. So you can say mobility, locomotion, or movement. So these two are having the same function. But there are some difference also between the cilia and the flagella. Structurally, both of them are same. Okay, if you see their structure, that structure I'll discuss. So structurally, both of them are same, but still some differences there. For example, cilia are more numerous in number and their length is short. So their length is short, but they are more numerous in number. Whereas this flagella, their length is longer, but they are less in number. Hardly you can see 1 or 2 or 3 flagella inside a cell. Okay, usually 1 or 2 maximum. Otherwise, the cilia they are very much in number, but their length is short. And this flagella, their number is less, but their length is longer. So this is the basic difference. Otherwise, structurally both of them are same. So the structure I'll discuss. So first you write down cilia and flagella are the hair-like outgrowth of the cell membrane. Hair-like outgrowth of the cell membrane. Okay, so structurally both of them have the same component. Both of them have the same components, but still some differences there. That cilia are numerous in number, but their length is shorter, while flagella are less in number, but their length is longer. And then what is the function? Both of them are concerned with the movement. Both of them are concerned with movement, locomotion, or we can say mobility or modality. Different words are used. Okay, so now we have to see their structure. Okay, so structure I will discuss from the data diagram is there in the NCERT. That diagram I will discuss here. So on page number 137 you can see this diagram. So this is the transverse section of the cilia and flagella. So this diagram is taken from this, especially this diagram is taken from the electron microscope, and this is the simplified diagram of this one. Otherwise in reality you will find this diagram. Clear? So this cilia and flagella, their outermost layer is composed of the plasma membrane. So the outermost covering, we can say the outermost covering of the cilia and flagella is called as plasma membrane. And their core part, this core part, apart from this plasma membrane, their core part is given a special name that is called as exoneme. That is called what? Exoneme. You can see it is highlighted here, exoneme. Clear? So this core part, exoneme, is composed of microtubules. Inside this exoneme, what is there? Microtubules. How many microtubules are there? So two types of microtubules are there. This you can see in the peripheral microtubules. They are called as peripheral microtubules, and they are present in the form of pairs. You can see each microtubule is in the form of pair. That's why they are called as actually they are present in the peripheral region, and that's why they are called as peripheral doublets. They are called as what? Peripheral doublets, because they are in the form of double, or we can say in the pair, and they are present in the peripheral side. That's why they are called as peripheral doublets. So this is one peripheral doublet. This is second one. So like this, total 9 are there. Clear? So in the exoneme, 9 peripheral doublets are there. And these 9 peripheral doublets, they are connected to each other with the help of this one. You can see they are called as linkers. Here it is given inter-doublet bridge, which is also called as linkers. Okay. So each peripheral doublets, they are connected to each other with the help of doublet. Then you can see 2 microtubules. They are single, they are not paired. So 2 single microtubules are found in the central. That's why they are called as central microtubules. You can see this is called as central microtubules. And both these central microtubules are also connected with the help of bridge. This is called as what? This one. This connection is called as bridge. And both these central microtubules are covered with or protected, we can say. They are covered with central sheath. You can see this red color central sheath is shown here. So it is present all around the central microtubule. Then I want to ask you whether any connection is there between the peripheral microtubules and the central sheath. Yes. So the peripheral microtubules, they are connected to central sheath with the help of a structure. You can see like a spoke-like structure that is called as radial spoke. So I want to ask you, how many radial spokes will be there? I think its number will be same as that of peripheral doublets. So just now I told you how many peripheral doublets are there? 9. So as 9 peripheral doublets are there, therefore the number of radial spokes also will be 9. And with the help of these radial spokes, they will be connected to the central sheath. Clear? So this is, you can see the pattern of the microtubules. So in the examination, one very famous question is there, that what is the pattern of microtubules in the cilia and flagella? So it is said that the pattern, or we can say the array of arrangement is 9 plus 2. That arrangement is called, you can see it is greener, 9 plus 2 array. So this 9 is what? 9 peripheral doublets are there, and 9 peripheral microtubules are there, which are present in the form of doublets, and 2 central microtubules are there. But the central microtubules, they are not present in the form of doublet, they are singlet. They are present in the form of singlet. Okay, so the pattern of arrangement of microtubules in the cilia and flagella is in the 9 plus 2 array. Okay, so this is the structure of the cilia and flagella. Clear? One more thing you should know, this cilia and flagella, they originate from a centriole-like structure. Okay, it is originated from a central structure which is called as basal body. Means we can say the origin of the cilia and flagella will take place from a basal, means centriole-like structure. That structure of centriole also I will discuss in the next discussion. So it will develop from the centriole-like structure which is called as basal body. So this is structure you may study from the NCERT. Okay, because it is very simple. Okay, so this one I explained from here. And that notes also you can prepare, that structure is given here. It is very simple. Okay, so that structure I discussed here, and that you can write from this one. Okay, so this is all about the cilia and flagella. From the next term I will start the centrosome and centriole. Thank you.